Hi dear students, welcome back to Sharon Zoology. In this session, I'm going to explain some previous questions from the chapter Molecular Basis of Inheritance. Then, Tyler conducted the experiment to prove semi-conservative mode of replication on. We know the semi-conservative method of DNA replication was experimentally proved by Merselson and Stahl. Merselson and Stahl experimentally proved the semi-conservative method of DNA replication. Franklin Merselson and Matthew Merselson and Franklin Stahl, they performed the experiment in 1958 to prove the semi-conservative method of DNA replication. Similarly, the same type of similar experiment conducted by, see here, the very similar experiment involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Vixia faba, the faba beans, by Tyler and its colleague in 1958. Okay, so Tyler and its colleague conducted experiment on Vixia faba or faba beans in 1958. Question: Tyler conducted the experiment to prove the semi-conservative mode of chromosome replication on which one? The answer is option B, the Vixia faba. Okay. Then the equivalent of a structural gene is option muton, second option cistron, third option operon, and the next option is recon. Let's see. What is structural gene? The structural genes are the continuous stretch of DNA that regulate a particular trait. The gene that continuous stretch of DNA that regulate a particular genetic trait. And cistron is a segment of DNA that coding for a polypeptide. See the statement from our NCERT book. See, look here, this is the structural gene. This is the smallest segment of DNA which undergo transcription. See, the DNA sequence coding for tRNA or rRNA molecule defined as a gene. However, by defining a cistron is a smallest segment of DNA coding for a polypeptide. The structural gene in a transcription unit could be a monocistronic or polycistronic. So the structural gene is similar to that of which one? Cistron, the smallest segment of DNA which is coding for a polypeptide. And structural gene is the smallest segment of the DNA which undergo transcription. So the structural gene is similar to that of cistron. Equivalent of structural gene is which one? Option B, cistron. So answer is option B, cistron. Which of the following RNA act as structural gene? Which of the following rRNA act as structural RNA as well as ribosome in bacteria? Which one of the following ribosomal RNA act as structural RNA as a part of RNA as well as ribosome? We know the 23S RNA. See here, the ribosome also act as catalyst. The 23S RNA in bacteria is the enzyme ribosome for the formation of a peptide bond. Let's see, bacteria has 70S ribosome. The 30S subunit has 60S ribosomal RNA and 50S subunit has 23S and 5S ribosomal RNA. Here the 23S ribosomal RNA has peptidyl transferase activity and catalyze the synthesis of peptide bond formation between the amino acids in bacteria. So this RNA also serves as structural component of larger subunit of bacterial ribosome as well as the enzyme ribosome. Thus the answer will be option C. Answer is option C, 23S ribosomal RNA. Answer is option C, 23S ribosomal RNA. The next one. A molecule that can act as genetic material must fulfill the traits given below except. See the options. It should be able to express itself in the form of Mendelian characters. It should be able to generate its replica. It should be unstable structurally and chemically. It should be provide for the scope for the slow changes that are required for evolution. Okay. The statement from our NCERT book. A molecule that can act as genetic material must fulfill the following criteria. First one, it should be able to generate its replica. Second one, it should be stable chemically and structurally. It should be stable chemically and structurally. Then it should provide the scope for slow changes like a mutation that are required for evolution. Then it should be able to express itself in the form of Mendelian characters. These are the characters shown by the genetic material. A genetic material must fulfill these following criteria. See here, it should be able to express in the form of Mendelian characters, true. It should be able to generate its replica, true. It should provide the scope for slow changes like mutation is true. Then it should be unstable is wrong. It should be chemically stable, not unstable. It should be chemically and structurally stable. So here the incorrect one is option C. So which one of the following is, a molecule that can act as genetic material must fulfill the trait given below except which one? Except option C. Next, DNA dependent RNA polymerase catalyze transcription on one strand of the DNA, which is called, which one? The template strand. See, DNA dependent RNA polymerase catalyze the transcription on one strand, which is called, which one? The template strand. The template DNA strand serve as RNA synthesis. Template is the strands which undergo transcription. The template DNA strands serve in RNA synthesis during transcription is called as anti-coding or antisense, while other strand is called as codings or sense strand. And its base sequence is same as that of the newly formed mRNA. So here the correct answer is option A, template strand. Look here, this is the template strand. This one is coding strand. This template strand is the strand which undergo transcription. So the DNA dependent RNA polymerase enzyme bind on this template strand. So answer will be option 1. Option A is template strand is answer. Next. Which one of the following is start codon? We know AUG is the start codon. AUG is start codon. Rest are stop codon. UAA, UAG, UGAR, stop codon. AUG is the start codon. Which of the following is required as inducer for the expression of lacoperon? Options are lactose, lactose and galactose, glucose and galactose. Let's see. Lacoperon means a set of genes that control the metabolism of lactose is called as lacoperon. So these are a set of genes that control the metabolism of lactose. See, what is gene expression? When will be the gene expressed in our body? When the cell need the product of gene, then only gene expression. See, in the absence of lactose, there will be no need of the enzymes like beta-galactosidase, permease and transacetylase. In the absence of lactose, there will be no need of beta-galactosidase, permease and transacetylase enzyme, because these enzymes are needed for the metabolism of lactose. They are needed for the catabolism of lactose into glucose and galactose. So in the absence of lactose, there will be no need of these enzymes. So there is no gene expression of ZY and A gene in the absence of lactose. But in the presence of lactose, see, in the presence of lactose, what happened? The lactose act as a inducer. In the presence of lactose, lactose act as inducer. Inducer inactivate the repressor protein. What will happen? the operator will be active. So the ZYA gene will be expressed to produce beta galactosidase, permease and transacetylase enzyme leads to the catabolism of lactose into glucose and galactose. So the lactose is the inducer in lac operon. So which one? The lactose is the inducer in lac operon. So answer will be option A. So we discussed only 13 questions from the chapter molecular basis of inheritance. Rest will be added in next video. Okay. So thank you for watching me. Bye.